Hi, everyone. Welcome to another webinar in our Researcher in Focus series. I'm very happy to introduce someone who is going to talk to us about something very interesting today. So our speaker for today is Dr. Jennifer Apu. She is a researcher at the Blue Economy Research Institute at the University of Seychelles, and she is going to be talking to us about impacts of seabird-derived nutrients on mangrove ecosystems. Okay. Hi, Jennifer.
Hi. Hello, everyone.
We're very excited to have you here today. I'm just going to share your screen to the stage now.
Okay.
I think you're already presented.
It's already. Okay, great. All right. So is this okay? Can you see my screen?
Yeah.
Presentation?
Yeah.
And you can hear me?
Yeah. You can hear
Yeah,
you.
perfect. Okay.
I'll remove myself now and then join back in.
Okay. Good afternoon, everyone. I'm very excited to be here. So thank you to the Indian Seabird Group for inviting me. I'm going to present to you today some results from my PhD. So for my PhD, I looked at the role of seabirds in nutrient cycling on tropical island ecosystems. And I'll be presenting today results from 2 chapters that looked at the impacts of these of seabird-derived nutrients on the mangrove forest. All right, so we all know that mangroves are these highly productive coastal forests that are found at the boundary between the land and the sea. And mangroves are very important habitats because they provide also a lot of ecosystem services and functions that are essential for us. So mangroves, they protect our coastlines from erosion. They serve as nurseries for numerous marine species. They provide essential resources such as fisheries and timber, and they absorb large amounts of carbon from the atmosphere, which contributes substantially to climate change adaptation and mitigation. And the provisioning of these functions and services, they're reliant on the health and the status of mangrove forests. But the health and status of mangrove forests are increasingly threatened by human activities. So mangroves, they thrive in low nutrient conditions, which makes them very sensitive to nutrient enrichment. And we know that human-derived nutrient enrichment may negatively impact mangrove forests, which affects their health and functioning, but also the essential goods and services that mangrove provides for us, as well as the surrounding habitat, such as surrounding seagrass or surrounding coral reefs. So we were interested in knowing that what happens when mangroves receive nutrients from natural sources. For example, nesting seabirds. So seabirds, they feed out in the open ocean and they return to their breeding colonies or their roosting colonies, where they deposit large quantities of guano. And this seabird guano is rich in nutrients such as nitrogen, phosphorus, and other essential elements. And we know that there is research that is increasingly showing that this nutrient enrichment by seabirds, they can enhance productivity around the seabird colonies, but also in nearshore marine habitats, such as coral reefs. Research has shown that on coral reefs, these seabird-derived nutrients, they increase fish biomass, they increase fish growth rates and coral growth rates. But seabird also use mangroves as a nesting and as a roosting habitat. And we were interested in finding out how this nutrient enrichment impacts the mangrove forest. So To do this, we went to Aldabra Atoll in the Seychelles, located in the western part of the Indian Ocean. So Aldabra has around 1,700 hectares of mangrove forest, which is the largest in Seychelles. Aldabra has also around 16,000 breeding pairs of frigate birds, comprising of both the great and the lesser frigate birds, and also a huge colony of red-footed boobies, around 35,000 breeding pairs of red-footed boobies. And on Aldabra, these seabirds, they nest exclusively in the mangroves. Okay, so to assess the impacts of seabird-derived nutrients on the mangrove forest, first, we quantified the amount of nutrients being deposited by breeding seabirds by sampling seabird guano and measuring the nitrogen and phosphorus content. We also sampled the mangrove leaves to determine if there was an uptake of these seabird-derived nutrients. We sampled the mangrove fauna consisting of gastropods and crabs, also to assess whether there was a transfer of seabird-derived nutrients throughout the mangrove food web. In these samples, I measured the nitrogen stable isotope ratios. So because of their high position in the marine food web, seabirds and their guano are enriched in nitrogen stable isotope ratios. So this is usually used as an indicator of seabird influence. So we measured the nitrogen isotope ratios of all these organisms. We also measured the sizes of the consumers to assess whether there was a link between the nutrient enrichment and the sizes of consumers. And then as well, we were interested in looking at whether there was an export of these seabird-derived nutrients to nearby coastal habitats. And for this, we measured nutrient concentrations in surface seawater in the lagoon, as well as also the macroalgae that were growing adjacent to the mangroves in the lagoon. And in the macroalgae here, we also measured the nitrogen stable isotope ratios. So we collected all these samples at 10 sites around Aldabra, comprising of 5 sites with seabirds and 5 sites without any nesting seabirds. And then we compared the nutrient status between these two groups. So first, the first result, using the seabird population size, the breeding metrics, and the guano nutrient concentrations, we found that seabird nutrient contributions to mangroves were substantial, with a combined total of around 40 tons of nitrogen and phosphorus deposited per year in the mangroves on Aldabra. So the frigate bird guano had higher content of nitrogen and phosphorus, but because the red-footed booby population is much larger than frigate bird population, the red-footed boobies had a higher nutrient contributions to Aldabra's mangroves. Then we looked at the nutrient content of the mangrove leaves. So leaves of the Rhizophora mangroves had higher nitrogen stable isotope levels at seabed sites compared to non-seabed sites. So remember I was saying this nitrogen isotope ratio is our indicator of seabed influence. So this results confirmed that there was a transfer of seabed-derived nutrients to the mangrove leaves. We also recorded higher percent nitrogen and percent phosphorus in the Rhizophora leaves at seabed sites compared to non-seabed sites. So nitrogen was around 39% higher and phosphorus was around 28% higher at seabed sites compared to non-seabed sites. So not only there was a transfer of seabed-derived nutrients, we also showed that there was an uptake of these nutrients by the mangroves. Next, using these foliar nutrient concentrations, we determined the nutrient ratios as an indicator of nutrient limitations. So here we found that the ratios of nitrogen and phosphorus were lower at seabed sites compared to non-seabed sites, which showed us that the uptake of seabed-derived nutrients by the mangroves reduced mangrove nutrient limitations to growth. We also, as an indicator of mangrove nutrient cycling, we also determined the resorption efficiency of nitrogen and phosphorus. So this corresponds to the percentage of nitrogen and phosphorus that is resorbed from senescing leaves prior to leaf fall. So usually when nutrients become available, mangroves will reduce their resorption efficiencies. However, for this one, we did not detect any differences in both nitrogen and phosphorus resorption efficiencies between seabed and non-seabed sites. Next, when we look at the macroinvertebrate food web, so this graph shows both the nitrogen and the carbon isotope ratios for all the samples, for all the organisms that we collected in the mangrove forest. So the most important result here, we see that we had higher nitrogen isotope ratios across all trophic levels. So this included in the baseline samples, which was our sediment organic matter, in our primary producers, which were the epiphytic macroalgae and the mangrove leaves, in the two primary consumers, which was the littorinid gastropods and the sesamid crabs, and also in the secondary consumer, which was the portunate crab. So this enrichment showed us that there was a trophic transfer of seabed-derived nutrients throughout the mangrove food web. Next, we look at the relationship of these isotope ratios, of these isotope values in the producers and the consumers. So the top graph shows the result. So it's a relationship between the nitrogen isotope ratios and their percent nitrogen content. So the top graph shows these relationships for the primary producers, and here we find positive relationships between the isotope ratios and the percent nitrogen, indicating that the enrichment by seabed-derived nutrients increase the nutrition of these basal food sources. And we can see that the effect was around 1.5 times higher in the sediment organic matter and epiphytic macroalgae compared to the mangrove leaves. For the consumers in the bottom graph, we also had positive relationships between the nitrogen isotope ratios and the nitrogen content, but this was only for our primary consumers, so the littorinid gastropods and the sesamid crabs, but not for the secondary consumer, which was the portunate crab. We also looked at the relationship between the nitrogen isotope ratios and the sizes of the two crab species that we had. And here we documented positive relationships between the nitrogen isotope ratios and the carapace width. For sesamid crabs at seabed sites, they were 6% larger compared to non-seabed sites. And also the portunate crabs were around 11% larger at seabed sites compared to non-seabed sites. So together, this indicated that the enrichment of the basal food sources, so usually crab growth is facilitated by the assimilation of enriched food sources. And here we show a link between this increase in crab sizes and the enrichment of the food sources, but also of the crabs. Then the last part that we looked at was the export of seabed-derived nutrients from the mangrove habitat. So we know that mangroves are influenced by tidal flushing. So there's constantly rising and falling tides that are constantly flushing the nutrients and the water in the mangroves. So for the seawater nutrients, I measured that during both the incoming and the outgoing tide. And here we found that the nitrate and nitrite concentration, so that's NOx, The concentrations was higher at seabed sites compared to non-seabed sites only during the outgoing tide, whereas the phosphate concentrations in surface seawater were higher at seabed sites during both the incoming and the outgoing tide. In addition to this, the macroalgae that we sampled adjacent to the mangroves, within 50 meters of the mangroves, they had enriched nitrogen isotope values at seabed sites compared to non-seabed sites. And together with these results of the seawater nutrient concentrations, this showed us that there was an export of seabird-derived nutrients from the mangroves to nearby coastal habitats, indicating that the influence of seabird nutrient contributions extended beyond the mangrove boundaries. So overall, in this study, we traced the transfer of seabird-derived nutrients throughout the mangrove ecosystem, and we showed that seabirds were maintaining this functional connectivity between the open oceans and the coastal seascapes. We also showed the role of seabirds in boosting the nutrient status of these nearshore coastal habitats. So by fertilizing the mangroves where they are nesting, seabirds are likely contributing positively to the productivity of the mangroves. And we also see that by increasing the sizes of mangrove crabs, which is an important component of mangrove fisheries, but are usually limited by catch size limits, seabirds, our results show that seabirds has the potential to increase crab fisheries productivity and benefit the provisioning of mangrove ecosystem services as well. So we conclude, therefore, that conservation and management actions should focus on restoring these seabird populations as well as their breeding habitats, such as mangroves, to increase this nutrient connectivity links and to improve the nutrient status of these mangrove habitats. So before concluding, I would like to thank all the funders and that supported this study, and also my supervisors that helped me with my PhD research. Thank you for joining and for listening to my talk today, and I'm happy to take on any questions that you may have.
Thank you, Jennifer. That was an amazing talk. So interesting. I think it's hard to just think of the role of marine predators in the ecosystem. I think this is such an important case, or like this is making a case for conserving them. It's really hard to imagine how connected everything is, but this really brings everything together. This is really, really cool research. I think as a layman, I can start by asking a question, but I think as a layman, I would like to ask, like, you collect all these samples. How do you process them? Like, how does it work? You go in, you collect the leaf, or you collect the guano, and then what happens?
Okay, so since I was interested in the stable isotope ratios, so one thing was important that you keep the samples cool during when you collect them, and you freeze them because I was working on a remote atoll, so I don't have access to a lab immediately. So I froze all the samples, and this stops any kind of chemical or biological activity that's still ongoing, and then I store them, and usually you have to freeze them at very low temperatures, -10, -20 degrees, and then after that in the lab you have to basically dry everything, so removing the water content in all your samples before processing it for stable isotope analysis. Yeah.
Okay. That sounds like really interesting fieldwork. We've also got a question from one of our viewers.
Mhm.
So they're asking, are there any other species that nest in the mangroves and can contribute to the nitrogen and phosphorus?
So for Aldabra, it's mostly these three species of seabirds: the great frigate, lesser frigate, and the red-footed booby, because they prefer these. They are tree nesters. They prefer this type of habitat. There are other seabirds on Aldabra, but they are mostly nesting on the ground, like the tropic birds, or nesting in burrows, like the shearwaters. Um, but in other, I guess in other places, there may be. I know in the Caribbean there are also other seabirds that nest in their mangroves. Seabirds that occur in the Caribbean. I don't know them at the top of my head, but yeah, I guess for Aldabra or the Western Indian Ocean, it's mostly the frigate birds and the red-footed boobies. We also have other types of boobies I know that nest in trees. Um, yeah.
And what sort of sets apart seabird guano from, like, I don't know if you call other bird poop guano, but what sort of sets apart seabird guano from other bird poop? Like, why are they so much more richer in nitrogen and phosphorus?
Well, it's because seabirds they feed on a marine prey, and marine prey are rich in proteins and they are enriched in these nutrients like nitrogen and phosphorus. So the seabirds, when they feed on these marine prey, they assimilate also these high nutrient values, and I guess that is what is transferred then through their guano um, when they are deposited around their breeding colonies. Yeah.
Oh, well, this was really, really cool. I think as a last question, because a lot of students watch our webinars as well, and it's
Okay.
a group that we like to target. Do you have any tips for anyone trying to make a career in this field in seabird research?
Uh, yeah, I mean, especially in the Indian Ocean, there's still a lot we don't know about our seabird populations. So I would encourage anyone who's fascinated by seabirds to go into it because it's really interesting and like now we're seeing all these connections that these ecological roles, these important roles that seabirds have. So there's still a lot we don't know. Like at the moment, I'm working at the University of Seychelles, and I'm extending this research to look at seagrass habitats. So is there connections between seabirds and seagrass? That would be an interesting question to see. So if you in an area where you have seabird breeding colonies and these seagrass, mangroves, or coral reef habitats, there's quite a lot we don't know about these mechanisms and processes of these nutrient links. But it's very essential because it informs us on conservation actions and management actions to restore these seabed populations, to restore these nutrient connections. And yeah, it's all about how ecosystems are connected. So it is a very interesting topic, and yeah, definitely go for it. If you have a question that you think it's worth investigating, yeah, you can extend it. Definitely, there's so much potential there.
Yeah. And sorry, one last question. What would you say the most important skills um, a person should gain to have a career in this?
Well, for me, field skills were very important.
Mm.
So, because yeah, also when you're on the field, you get inspired and motivated because you make all these observations. You understand your research. So
Yeah.
field skills always come in handy, uh, and it kind of yeah, it's the driver I would say behind any research. Is like being in the field and observing seabirds or any behaviors or processes that you may be able to observe. Yeah.
Oh, thank you so much for joining us today, Jennifer. if anybody watching this later has any questions for Jennifer, please feel free to post them on our um, YouTube comment section, and then we can reach out to Jennifer and get them answered for you. And I hope you enjoy watching this talk because it's so interesting. Thank you.
Thank you so much for inviting me. Thank you, everyone.
Thank you.
Bye.